Hello, everyone. I'm Lucia Gambuza with Modern Retina, joined by Dr. Mark Barakat, a vitreoretinal surgeon, director of research at Retina Macula Institute of Arizona, and an investigator in some of the most closely watched trials in the field right now. Thank you for joining us today.
Thanks for having me. Pleasure to be here.
Okay, today we'll be discussing the recent clinical trials at the Summit Conference in Las Vegas that occurred from June 12 to 13. Dr. Barakat just presented data from the SOL-1 Phase 3 superiority trial of OTX-TKI, the first trial of its kind to demonstrate superiority over an approved anti-VEGF therapy in wet AMD. We're going to dig into those results, what he's taking away from this year's program, and where he thinks the retina field is headed. Let's jump right in with our first question. Dr. Barakat, CTS brings together a unique mix of stakeholders. From where you sit as both a researcher and practicing clinician, what does that cross-sector conversation add to how you think about the trials you're running?
Honestly, CTS is one of my favorite meetings. Not necessarily about the innovative program, which it is, but also all the hallway conversations. So here you have the folks that helping with the logistics, the CROs. Then you have the reading centers. Then you have AI, you name it, right? You have regulatory conversations. You have the clinical trialists that are there. You have the sponsors that are there. You have pharma that is there. You have investors that are there. So you're looking at it from if there's an angle of looking at drug development, it's being covered, and so it's great to learn from peers and mentors, and it's just a fantastic a mix of things that are important for clinical trials.
Yeah, absolutely. And I love that you spoke about that diversity of the stakeholders that are there. And in this year's program specifically, there were a dozen new panels specifically based on attendee feedback from previous years. AI, as you mentioned, regulatory affairs, partnering with large pharma. Were there any topics on that expanded agenda that you felt were overdue for this kind of dedicated forum?
So one of the things that I need to learn more about, so a personal weak spot for me is trying to get a better understanding of regulatory. Right now, we're all wondering about, well, can you get an approval from the FDA with one really well-controlled, highly statistically significant trial? Does it need to be powered for safety? So the conversation that included Wiley Chambers were actually really, really interesting to me. So I think that would be one of those things. The other things that I always enjoy, and we've had panels like this before, but every year it feels like it's growing at a faster pace, is what AI can bring to the table in terms of finding the right subjects, in terms of helping to reduce screen failures, but also in terms of helping just generally the flow of clinic and clinical trials.
Do you think other colleagues of yours share the same sentiment?
So I mean, the short answer is yes, because I think everybody in the room was enthralled by these
Yeah.
presentations. Now, it might be a little bit of an echo chamber in the sense that, again, this caters to the crowd that is very vested in clinical trials. But that's what makes this so interesting to us, because we're all rowing in the same direction. We see the potential of many of these compounds. I mean, what does it take to efficiently get these to the finish line? And frankly, that was part of the discussion too. If we can increase efficiency, then perhaps we can get more or better agents, more diversity out there as well. So it's fascinating.
Let's shift gears to the SOL-1 efficacy outcomes that you just presented at the CTS audience. Can you start by giving us the headline results and then help us understand what made this trial design so significant?
Yeah, I'm glad you asked. So headline results are this. This is the first trial in, well, a long time that I can recall that is not a non-inferiority trial. This is a superiority trial. So basically, subjects with naive wet AMD were randomized in a one-to-one fashion. They all had a monthly aflibercept run-in, and they were randomized one-to-one to receive 2 milligram aflibercept versus OTX-TKI, followed until the primary endpoint was at week 36, and then the final readout was at week 52. And really what you were looking for is these patients, these eyes had to have actually quite excellent vision to be randomized. At randomization, they must have gained either 10 letters or essentially achieve 80 somewhat letters, so about 20/20. So these are very, very well-seeing eyes. And what you're looking for is you're looking for a loss of 15 letters in order to receive supplementary treatment. Why 15 letters? Because again, we're talking about regulatory again, right? This is FDA. Under FDA guidance, 15 letters is significant. Why do a head-to-head with one aflibercept versus one OTX-TKI at randomization? Because you don't want any sham treatments, and you want to have the same treatment schedule. So these are all based on the draft guidance that the FDA has put out, and under SPA treatment, on a special protocol assessment. Right? So what's the end result? The end result at month 9, at week 36, is that close to 75% of eyes that had received OTX-TKI were rescue-free, that maintained vision as defined by not losing 15 letters. But that's the primary endpoint. If you actually dig a little deeper, look at all the secondary endpoints. If you look at the anatomy, which is what we treat in clinic, the anatomy showed a large degree of stability in terms of volume. If you look at the time it takes for the anatomy shift by 30 microns, which we know has been associated with fluctuations that could be detrimental to the vision in the long run. So a 30 micron shift, it takes 23 weeks longer in the OTX-TKI arm than in the aflibercept arm. And the same thing in terms of when you get your first rescue treatment, that's 24 weeks longer in the OTX-TKI arm. So basically, you get shifted by approximately 6 months. So here we have an agent that shows the potential of durability up to 9 months. And that's actually very exciting, especially giving a clean safety.
Absolutely. Those numbers are really compelling. And what's especially interesting is what they mean beyond the data. So let's talk about what this looks like in the real world. For a
Mhm.
patient sitting in your chair, you mentioned rescue-free rate findings. Could you elaborate on that and what those numbers actually mean for the patient? Are those patients genuinely coming in less often, or is the clinical picture a little bit more nuanced?
It's always going to be more nuanced, right? Of course, there's an intersection between clinical trials and real world, but that intersection, you have to define once you get it into a different mixed patient population, right? So, would I in clinic necessarily wait for a 15-letter loss prior to treatment? Probably not, right? Flip side is I might just look at the OCT findings and go from there. Will it be? I do think it will shift the paradigm. And this is why I think that. If I have a patient that comes in with your typical bolus injections, and they come in at whatever interval that we've designed, and I see subretinal fluid or intraretinal fluid that wasn't necessarily there last time, or perhaps was shifting a little bit, they get another treatment. End of story. However, if I have something that has shown activity, that has shown proven activity over the course of 6, 9 months, and I see some fluctuations in fluid, I may decide to monitor them for now because I know there is treatment on board. So I think we as a community, assuming approval, we'll have to figure out how to implement this in our clinics. And I think we have to learn not to be as trigger happy as we were before. I think, well, that's a loaded term, but I think it's appropriate if you have bolus therapy. But now we have extended therapy, and you could perhaps layer the bolus treatments on top.
And I think a big part of why patients are also willing to consider something new comes down to trust in the safety data. And we can talk about that now. The safety profile of Sol 1 was notably clean. As someone who counsels patients about ophthalmic implants, how do you approach that conversation with a patient who's already been on a monthly anti-VEGF injection and might be skeptical of anything new going into their eye?
So, well, here's the thing. I always tell patients whenever we switch to new therapy, you always worry about safety, 100%. So things that you worry about is inflammation. Well, there were some mild to moderate cases of inflammation that resolved with topical therapy, some frankly without. So actually really well tolerated in that sense. There were no cases of occlusive vasculitis, things that would be necessarily a game changer, right? You don't want to inject something where you have a fear of permanent loss of vision. Now, of course, you're going to see the same things you see in most trials. You see subconjunctival hemorrhage, things of that nature. You do see some cataract formation, but that's in keeping with other trials. Floaters do happen. This is an implant. It's an implant that dissolves. But frankly, if you look at the floaters and if you look at the visual acuity, the visual acuity was actually quite well. So I think all in all, my conversation with patients would be, here we have an agent that has a potential to last 9 months, possibly longer. Because frankly, in the real world, I might do titrate that as well to my liking, maybe 6 months, maybe a year, you don't know. It has potential to last longer. It does have a different profile than the other injections. One of them may be floaters, but I have confidence that those floaters, based on the delivery technology, will disappear. So I'm not worried about that. Actually, you could almost argue, you could almost posit this. We're going to inherit a entire cohort of patients in retina clinic that dislike floaters. We even do vitrectomy for floaters. And the irony is, by injecting these patients, say monthly or bi-monthly every 3 months with your regular agents, you'll get little oil bubbles, you get little floaters, and we are actually introducing more permanent floaters with these bolus injections than we might with an injection where I know that they will dissipate. So I can see it any which way, and I think it's just nice to have that optionality.
When the field eventually has multiple approved TKIs to choose from, do you think we'll be differentiating them on mechanism, delivery system, duration, safety? What's the variable that will actually matter to a practicing retina specialist?
That is impossible to tell. Obviously, whatever you feel most comfortable with, right? So if there are no head-to-head trials, you will not see this, right? One of those things will be the patient experience. So we talked about floaters. Here we have a platform where I know the hydrogel will completely dissipate. So I don't have any long-term concerns. Assuming that safety is clean all the way around, of course, otherwise we wouldn't even be having this discussion. And then really what it boils down to is, do you have an agent that's superior and maybe will allow me earlier access? So that's another thing that we discussed, which is interesting. We live in the world of step therapy. We don't necessarily want to, but that's the world in which we live. And technically, you can make you can take the position that here, this is the first time you've shown superiority to aflibercept 2 mg. So I can now confidently approach a payer in a conversation, say, why are you blocking me from using this? This falls outside the step therapy. Now, these are all interesting angles. I think it's going to depend on every person, every clinician.
Let's bring it back to the CTS conference. What was one presentation or moment that genuinely surprised you or you found most compelling or made you think differently about something?
It wasn't necessarily one presentation. What I enjoyed the most from a personal level was the panels on artificial intelligence. And I know AI is very hot right now and will continue to be so, but I'm always surprised at how far we've come in terms of, if could you go back 5, 10 years and ask a wish list of a clinical trialist, say, well, what if I told you I could come up with a program that looks at OCTs for you, segments it for you, then correlates it with visual acuity and other inclusion exclusion criteria, and then just gives you a list. And I would laugh because basically you're saying, do I get a pony with that too? But now we have that. And not only do we have that, but we have multiple different versions and approaches to it. So I'm it continues to amaze me, and kudos to all the companies in AI, and keep up the good work.
This is a beautiful transition to my last question, and it's more of a bold prediction for you. 5 years from now, what does the standard of care look like for a newly diagnosed wet AMD patient walking into your clinic? Is anti-VEGF still the anchor?
No, absolutely. I think anti-VEGF is here to stay, but I think 5 years from now, I can completely see myself an E patient coming in. I'll layer on a tyrosine kinase inhibitor, so I'll put on OTX-TKI. I will monitor the patient, have them come in for OCTs, or maybe even put them on home OCT, right? And just kind of monitor the fluctuations of the fluid for now, figure out what the pattern is, and have them come in every now and again, maybe every 3 months, maybe every 6 months. If at the time home OCT is problematic for some patients for whatever coverage, maybe even have them come in clinic to just get their OCTs done and just do an OCT clinic and just call them back saying, well, you have a little bit of fluid, maybe you should come out in a few weeks for an injection. Why no longer the Why no longer the urgency, you ask? It's because I already have something on board treating it underneath. So I have this extra margin of a safety margin that's already in place. And so I think that's the sort of the 5 year. In 10 year, not only will you be looking at that, but you'll also be, how do I introduce, I mean, I think home OCT at that point will be firmly entrenched, but how do I then introduce gene therapy into the mix? Because that's also really enticing for very similar reasons. So it's a fantastic field. I'm very excited, as you can tell, which is why I think CTS is really crucial and help us drive and keep the momentum going in terms of bringing these new developments to the field.
Dr. Barakat, thank you so much for your time and for sharing your insights with us today.
Thanks so much for having me. Pleasure.
And thank you for watching our Clinical Trials at the Summit 2026 conference coverage. Visit Modern Retina for more on the latest news and conference coverage in the retina world.